Okay, so this is my fifth lecture, and as I said right in the beginning, this is the one picture that has been constant throughout my lectures, and this sort of illustrates this relationship between the plant world, the animal world, and human beings, and also illustrates the point that a lot of the diseases that we worry about now are diseases that are not sort of really human diseases. They're diseases that come to us typically from the animal kingdom, from bats or from monkeys, birds, etc., etc., etc. And that's because typically there are viruses that circulate in these creatures and don't do them harm, but they can reassort or combine to form viruses that then become infectious to human beings. And because these are novel to us, we have not encountered them before, the question of our immune system's ability to deal with them becomes that much more complex. And typically we will have immune problems. So when H1N1 came around, that was a new disease as far as humans were concerned. We had no or very little pre-existing immunity to that disease at that point when it came. So one question is, can you predict where these diseases will come from? Where will the next emerging infectious disease come from? Is it Nipah? Is it something else? What it might be? And for that, and also why are we seeing this increase in these special emerging diseases that are zoonotic in character, that are called zoonoses because they originate in animals? So that's partly because as human beings expand their region of influence, we drive wildlife into ever smaller areas. So there's an ecological component to why this happens as well. It's important to understand that these are complex issues. So today we've done all of this. Today what I want to talk about is vector-borne diseases, network and agent-based models. And there's a tutorial we'll spend some time on, but this is the link for the tutorial. So just please write it down exactly as it is. It's I is capital, D is capital, T is capital. So it's infectious disease tutorial.pdf. And you take your laptops out later, you can try and access this. I can also write it out here. Okay. So let's just revise what we did the last time. We talked about structured populations. We talked about the SIS model for sexually transmitted diseases and the existence of a low risk and a high risk group. We stressed this WAIFW matrix, this who acquires infection from whom matrix. We talked about the slaving of dynamics in both groups at long times, age structuring. Then we talked about childhood diseases and age classes C and A. And then we moved on to doing things like metapopulation, migration, commuters, etc. We talked about stochastic models and how to make a model stochastic. So one way that we started out was to take the original ODEs and add terms to them that were different at different times, that represent some sort of unknown or random feature that was added to these equations. Okay, by adding what are called noise terms or stochastic terms. We said that the moment you introduce stochasticity, various issues of critical community size arise. If the number is too small of the people in the community, people can die off or recover before they can infect someone else. So the fact of what is the critical community size is something I mentioned very briefly and said that this is important in the context of stochastic situations. These can lead to extinctions for small communities. Then we spent some time talking about in the Poisson distribution, the exponential distribution of time between events, the fact that the time between events, if it was exponential, that indicated that the process was memoryless. It doesn't depend upon where you start. Then we talked a lot about how to draw from a specific distribution. I showed you this trick of taking the inverse of the cumulative distribution function and relating that to a number uniformly drawn from 0 to 1. Then after having done that, I talked about spatial models with diffusion terms, related this to the 1D Fisher-Kolmogorov equation, which are traveling waves, gave the example of rabies. And then we finally finished up by talking about cellular automata models, in particular the forest fire model that had the same structure of waves of infection, bordering susceptible, and in that case susceptible, infected, and then recovered. Okay, so I did this a little fast, but you will probably see this in the notes. So the idea is that most of us will put our notes up on some common web page, or at least the slides up on some common web page, which will be password protected or password access, and we'll give you the password so that you can have a look at it at some later point. So then you can go through these arguments, and if you find errors in them, you can point them out to me so that I can correct them in any subsequent version of this talk. The one thing that I wanted to go over again, partly because it's part of your tutorial, is this question of the time distribution between successive events for a Poisson process. Okay, so if the event occurred at time t, the probability p of tau that the next event occurs between t plus tau and t plus tau plus dt is a decaying function of this form. If this process has some rate a, okay, it has that particular form. This form reflects an exponential distribution, which means it's memoryless. So this is for 3 different choices of a. This is what that exponential actually looks like. The larger the a, the larger the rate, and the sort of faster this falls off over here. When we talked about stochastic disease models, we sort of tried to enumerate the different things that could happen. And immediately you see that one of a large number of possible events can happen. Each is independent of another event, each is a Poisson process with its own rate. So here are the number of events that could possibly happen. If you start with some initial number of S, I and R, suppose you have 19 S's, 20 I's and 3 R's. That can go to a situation where the S goes down by 1, keeping the infected and the recovered the same. So that is someone dying from the S compartment or being removed from the S compartment directly. I could have a situation where SIR goes to S minus 1, I plus 1, R. That is someone being infected in the S compartment and moving to the I compartment here. Also, I could have SIR going to SI minus 1, R plus 1, which is someone leaving the I compartment and going to the recovered compartment. If I have 50 individuals, 100 individuals, each of this is a rate for each of those individuals that I have to worry about. So now the question is, if all of these things can happen, all of them can happen independently, when does the next event happen and what is that event? How do I calculate that? So I'm going to think about this from the point of view of having all of these things that can possibly happen, but it's my job to determine in a computer simulation which is the next event and when does it happen. Okay. So I did this somewhat fast, but here I just want to do it a little slower so that you understand it better. This is what is called a superposition of Poisson processes. Suppose I have 2 processes, N1 of t and 2 of t, that are independent and happen with rates A1 and A2. So this is time going forward in this direction, and each of this is the first process happens here, first process happens here, first process happens here, here. And inside that, or tied up with that, is second process happening here, second process happening here. Okay? So instantly you can see that the first process happens more often. Therefore its rate is larger. The second process happens much less often and its rate should be smaller. Okay? So now what I want to ask is, I can make one process by just taking the union of these things, sort of coloring them all the same color, which I have done here. Okay? So by doing this, I don't care which process, I just want to know when the next event, which could be either this or this, actually happens. So now the idea is that given the rates at which A1 and A2 at which this happen, this process is governed by rate A1 plus A2. Okay? And now I can ask, when does the next event happen in this case? So the time to the next event is exponentially distributed with the rate equal to the sum of rates over all possible events. In that example I gave you 2. I said 2 processes, I1 and I2. The rate at which the combined process happens is A1 plus A2. If I have a large number of processes, I have to add up all the A's which I have done here. So that distribution function now is this exponential of minus tau times sum over all I AI, and the sum over all I AI appears also there. Okay? So this is finally some number and some number that reflects the sum of rates. Now I know how to do this because I figured out yesterday how to draw from a particular probability distribution. This is to figure out when does the next event happen by drawing from precisely this distribution, A e to the power minus A times t. Okay? How can I answer which event occurs next? For example, here I can say that look, it's more likely that an event of this type will occur sooner then something like this, just because the rate is so much smaller for the second event, whereas the rate is much larger for the first event. The way that's done is to just weight these by their actual rate. So this is the sum. The probability that a particular event happens, called V happens, is the rate for that V event to be happening summed over all the rates. So in that example there, the much more likely event, the one with a much larger rate, figures much more here. Suppose that happens 90% of the time. The ratio of the rates is sort of 9 to 1 or 90 to 10. Then if I add those rates up, it will be 100 here. So the probability that the first event I1 happens is 90 over 100 or 90%, and the probability the second event happens is 10%. Okay, so that's how I determine which one is actually going to happen. So here, all the weights are positive because these are rates. Rates cannot be anything other than positive. So a probability of specific event is given by this weight. The probability that the next event will happen at a time tau is given by this. We don't know what event it is. One event is going to happen. So there are two things. First is determine which event is when an event is going to happen. Second is determine which event is going to happen. Okay. So the way we think of implementing is set initial population number, 1000 people, 500 people, 10 people, etc. That's your starting point. That's your t equal to 0. Then you calculate all the rates for all possible things that can happen. In that particular example, only two things can happen. Either there is a blue event or a red event, or a green event, or whatever the color was. So calculate those rates. Then you choose tau from an exponential distribution with this parameter, sum over I, ai, as in this here. Okay? That is by the same trick of inverting the distribution, of taking the cumulative distribution function, inverting it, finding out what is the value that corresponds to a uniformly drawn random number distribution between 0 and 1. Okay. After that, now you've decided what the time, when is that event going to happen. The second decision is which event is going to happen, and that's determined by choosing it in proportion to this or to this. So 90% of the time I choose the first event, 10% of the time I choose the second event. Once I have done that, I change the number of individuals to reflect that particular event. So I do remember which event it is that I'm choosing, and I make the update. So now S can become S minus 1, I can become I plus 1 if I've chosen that event. Maybe S becomes S minus 1 if I've chosen that event. All of these events are independent of each other. So they can all be added up in this particular way. Once I do this, I go back to step 2. So I just wanted to do this in some detail because I wanted to make sure that you understood this. Here's a little small detour about control of disease. And that goes back to our earlier discussion of what R0 was, what the basic reproductive ratio was. So this has three parts to it. One part is how long do you remain infected. One part is what is the rate of contacts between susceptible infectious individuals. So rate of contact into duration of infectiousness is number of contacts. Out of that number of contacts, what fraction is successful in transmitting disease? So there are these three things that go into the definition or the way we think about R0. And that's really what it is. I can contact 25 people. There is some probability that one of these contacts actually is an efficient contact, in the sense efficient in transmitting disease from me to you. And that's really what R0 is. How many secondary cases are there when there is one person infected in a completely susceptible population? But now that I have split this up, this gives me a hint as to how you might control a disease. So the first way is to reduce transmissibility. Okay, so that is this point here, that is this point, the tau. If I reduce tau, for example, if I deliver vaccines or use antiretrovirals reduce the viral load, the number of viruses in your bloodstream, down to a low level. Okay, so that makes it that much harder for it to communicate to another person. The second is to decrease your mean contacts, that is this part here. And the way that is done is to isolate people or quarantine people. And also give them health education programs. And the most important health education program is if you fall sick and you have the flu, which is likely to be infectious, just stay at home. Stay at home, drink water, don't talk to too many people. You will recover automatically in 3 to 4 days because that's a standard, it's a viral disease, it's self-limiting. Okay? The third part is this part, the duration of infectiousness. And that you can limit by therapeutics, antibiotic treatments, anything that boosts your innate immune response will all help to make you better faster. Okay? So all of public health interventions really rely on, given this idea behind what determines R0, behind doing all of these things, maybe some more things. So that's where thinking about diseases from a theoretical point of view is important because it helps you focus on what are the different components that determine how efficiently a disease is spread to another person. And you can decide that, look, it's hard to do this, but maybe I can do this. I can encourage people to isolate themselves. Maybe I can do this particular thing. So exactly the right mix of different ways of dealing with the disease have to be decided by some sort of public health authority or people who think about this problem. Okay, so now I'm going to sort of describe a vector-borne disease to you. That's going to be really descriptive because there's going to be you have to understand the background. There's not going to be much modeling that I will talk about because that will be a whole separate set of lectures on their own. But it's important to understand what is the problem about vector-borne diseases and how do they differ from the directly transmissible diseases that we have been discussing so far. So malaria and related diseases, it's known that mosquitoes, a very specific species of mosquitoes, transmit pathogens, pathogenic organisms, typically unicellular organisms or viruses that cause malaria, filariasis, dengue, yellow fever, West Nile fever, Rift fever, as well as a number of others. Okay? The idea that mosquitoes would be responsible for transmitting a disease goes back to Manson in 1877, working in China, who showed that mosquitoes transmit some pathogen that is borne by blood. Okay? Then, some years later, Laveran actually observed the malarial parasite under a microscope. Ross in 1897 showed that mosquitoes transmitted the malarial parasite and worked out the whole life cycle of the mosquito, of the parasite within the mosquito. In 1900s, these people showed that mosquitoes transmit yellow fever virus in Cuba. In 1906, it was shown that mosquitoes transmit the dengue virus. Okay? Here's how you know if you have malaria or not. How many of you have had malaria? Nobody. Okay, that's pretty good. But anyway, so now you can recognize if ever you get it in the future. Typically, the main symptom is a sort of shaking chills that happen usually in the evening or at a fixed time in the day. High fever, lots of sweating, headaches, nausea, vomiting, diarrhea, anemia, muscle pain, etc., etc., etc. So, a lot of these are typical to a lot of viral diseases. So the only real diagnosis of malaria is if you know that there's a lot of malaria going around, and as well as a blood test that you can take, which is a fairly straightforward test, you can figure out. And of course, you don't want to get into the situation where you have a coma or anything like that. But most cases fall in the upper limit here. These are the countries that exhibit malaria in endemic form. Notice that pretty much they're confined to an equatorial band here. And that's because mosquito prevalence, etc., relies really, the ability of mosquitoes to transfer infection, especially also, relies on the fact that mosquitoes tend to breed in more warm, more humid climates. Okay, you can see that Africa is fairly well distributed. There's some India, this is Burma here, which is again a hotspot. Parts of Southeast Asia, parts of these, so this would be, I guess that would be Indonesia sitting there, and parts of South America right up to Mexico, further up. In 2015, about half the world's population was at risk of malaria. And most of these cases were in sub-Saharan Africa, that is this part here. This is the Sahara, sub-Saharan Africa is the part that comes below that. In 2015, this was home to about 90% of malaria cases and 92% of deaths. And the WHO estimated 212 million cases of malaria in 2015 across the world, and close to 500,000 deaths arising as a consequence of malaria. So the malarial parasite infects two types of hosts. It infects both the mosquito and it infects the human as well. It doesn't make the mosquito sick as far as we can see. It doesn't probably affect the lifespan of the mosquito very much, but it makes the human being fairly ill. So the mosquito is a particular mosquito. There are a very large number of mosquito species, probably in the region of about 3,000 different species, but only a small number of them are involved in transmitting disease to human beings. And the mosquito here is called the Anopheles mosquito, and it's called a vector because it transmits disease. Pretty much most vector species bite between dusk and dawn. So early in the morning, sort of in the evening between 6 and 8 or 9, as you know, is the time when you tend to get bitten most. The intensity of transmission depends upon a number of factors, right? Parasite, vector, host, environment, etc. It's the epidemiological triangle with the introduction of a vector component to it as well. So I say there are about 3,500 species of mosquitoes, so that's what the Anopheles mosquito look like. Human malaria is transmitted only by females of the Anopheles genus. Of about 430 different types of Anopheles, only about 30 to 40 transmit malaria. So they act as vectors. So it's not all the mosquitoes. There are a very large number of mosquitoes involved, but only a small number of them are involved in transmitting disease, function as vectors. If at some point any of you go to Pondicherry, there is a vector research center in Pondicherry that has a room inside. It's called a mosquito museum. So there they have catalogued various species. They have little samples of large numbers all mounted and so on. I think samples of mosquitoes caught from pretty much every area in India, maybe some even from outside India. So I think they probably have 400 to 500 species caught from around this neighborhood. Yeah. These are some indication of different types of mosquitoes, of Anopheles mosquitoes. So Pulcherimus, Messae, Melas, Nistus, etc., etc., and what their distribution is around the world. In India, it's Anopheles stephensii, fluviatilis, minimus, Pymai, stephensii again here, this one, and something here in the Andaman Nicobar Islands that seems to be peculiar to that. But the main one is Culifaces over here, which pretty much spreads out over the whole of India. It doesn't really matter. These are sort of differences in the nature of, these are of interest to a naturalist, but they all sort of transmit disease pretty much in the same way. Okay? Alright, so let's talk a little bit about the mosquito itself and why it's so interesting. The mosquito life cycle starts off as an egg. This egg is typically laid on the surface of water over here, and then the egg matures and becomes a larva. It breaks open, becomes a larva. The larva becomes a pupa. So all of these stages are still at the surface of water or underwater. So these are called aquatic stages, and they last anywhere between 5 and 14 days. Finally, this becomes something that can fly out of the water. So this becomes an adult, and it's a female adult Anopheles, which is a malaria vector. How long does the mosquito live? That turns out to be a fairly controversial question that we've also been trying to ask at various times to get number for. Well, they can live up to about a month, but depending upon whether if you look at them in lab situations or whether you look at them in the natural world, there are differences to these. And probably in the outside world, adults probably live somewhere between 1 to 2 weeks out of very specific protected lab contexts. Once female mosquitoes are ready to bite people, they seek what is called a blood meal. This is the technical term, which means they want to bite you and suck a drop of blood out of you. So all of you who have hit mosquitoes like this, to sort of know that there's a lot of blood, that's your blood that they have just been sucking on. It's called a blood meal. Each species of Anopheles has its own specific habitat that it likes. Some like dirty water, some mosquitoes prefer clean water, fresh water, etc. At a time, somewhere between 50 and 200 eggs are laid out at a time. They're laid singly on water. These eggs, it doesn't matter if the water dries up, because the egg the egg is not resistant to drying, which means it needs the water, but they hatch within about 2 to 3 days. So after sort of a full blood meal, the female rests for a few days while the blood is digested, and at that time the eggs are developed. This depends upon what temperature it is, but usually about 2 to 3 days in tropical conditions. So here again is that cycle here. So once the eggs are fully developed, the female lays them and resumes seeking a host again and can go back and do this. So the cycle repeats until the female actually dies. Transmission tends to be more intense by places where mosquito lifespan is longer. That's because the parasite has time. The parasite has a life cycle of its own inside the mosquito. It has a life cycle of its own inside the human host. So if the mosquito dies very fast, there's not enough time for that life cycle of the parasite to develop within the mosquito. And this is what sort of, if these mosquitoes feed, all they want is blood. So they're perfectly happy to feed on cattle or other animals. But if they have a preference for human beings, that means that they're a little more effective in transmitting the infection to human beings than to other animals. So both this long lifespan and strong human biting habit of the African vector species is really the reason why most malaria cases are found in Africa. There are some other reasons, but that's certainly one strong reason. So transmission is usually seasonal. It peaks during and just after the rainy season. And you see epidemics when climate and other conditions favor transmission. For example, rain, stagnant water where eggs can be laid. We favor transmission in areas where people have little or no immunity to malaria. And also when people with low immunity move into areas with intense malaria transmission. So that could be refugees who come from elsewhere, come up all crammed up together in places where hygiene is low, where water can remain stagnant. So that's another reason why you can have a sudden spread of malaria cases. That's what, so these are red blood cells. So this is looking at a red blood cell, and these are the infected red blood cells that you can see here. So the malaria parasite is parasitic, it's a protozoan, a single-celled organism. It's called Plasmodium. You probably remember this name from school, Plasmodium, the malaria parasite. There are more than 100 species of Plasmodium, and 4 were recognized fairly early to infect humans in nature. So there's Plasmodium vivax, falciparum, oval, malari, and fairly recently, since 2008, about 10 years ago, a new species of Plasmodium is known to infect humans. What happens in humans is that the parasites grow and multiply, first in the liver cells and then burst out of there to enter the blood. So they enter the red blood cells. So what you see here are red blood cells, and there's a parasite, there's a parasite. In the blood, broods of parasites grow inside red cells and destroy them, breaking them up, releasing them, so the daughter parasites can go and invade other red blood cells. Okay? It's these blood stage parasites, so it's not so much when it's in the liver, but when it's released into the blood, then it causes the typical symptoms of malaria. So here is the sequence. This is where do we start? We start with the human being who is infected. The human being is bitten by a female adult Anopheles mosquito. That the Plasmodium, the single cell organism, develops within the mosquito. Then the mosquito bites the host. After that, this organism enters the liver of the host. From the liver, it is released into the bloodstream. From the bloodstream, it breaks up these cells and then releases sort of further microorganisms once it sort of multiplies, and then leads to the progression of infection in that human being. This is that expanded a little bit to sort of show you how complicated the whole life cycle, both within of Plasmodium, both within the human and the mosquito is. So it comes in here, attacks a liver cell, a hepatic cell, goes through various complicated stages here until it becomes mature. It breaks out, finds a red blood cell and attacks it. Within that, it has a complicated life cycle again until it reaches something called a schizon state. It breaks up and then begins to infect further red blood cells here. And at this stage, you can either have female or male versions of this. This is then picked up by an Anopheles mosquito biting a human being, and this is the cycle within that. So it moves from the midgut of the mosquito to the salivary glands of the mosquito, and from the salivary glands is when it's actually pushed out when a mosquito bites. If it was in the midgut, it wouldn't matter, but it's part of the saliva that is released when the mosquito actually bites a human being. So much is known about this, as you can see. There's a lot of work that has gone on, a lot of basic zoology and biology, and of course malaria is among the most intensely researched of vector-borne diseases, precisely because it is such a big public health problem. Okay, so much research actually goes on to figuring out where can we stop this multiplication, at which stage is it useful to attack the malarial parasite using a drug, what is the efficacy of the drug here, as well as there are other public health measures that you can do on this side. Okay, what are the sort of variables that we might need to describe? So one variable here is just the population density of humans, which must surely be important. So call that H. Population density of mosquitoes, yes, because mosquitoes bite, call that N. For simplicity, we'll just say mosquitoes are all female. So we'll just consider this to be the female mosquitoes. The number of infected humans is X. The number of infected but not yet infectious mosquitoes is Y. And of course, the number of uninfected humans is just the total population density. So let's call everything a density rather than a number, just to be consistent with this. We need to worry about the human blood feeding rate, that is what proportion of mosquitoes feeds on humans every day. We need to take into account the survival of the mosquito. What is the probability that it survives a day? or a probability that it is sort of dead at this point. We have to worry about the behavior of the parasite inside the human, inside the mosquito, the number of days from infections from the mosquito, what is the proportion of infected humans that are infectious, that is, probability a mosquito becomes infected after biting an infected human. There are a whole bunch of variables that are there that you in principle need to worry about. From this you can make useful combinations. If you divide the number of cases by the total number of human beings, that's the prevalence of malaria, the number of cases at a given time per unit of population, which is x divided by h. The fraction of infected but not infectious mosquitoes is y divided by the total number of mosquitoes. The fraction of infectious mosquitoes is z divided by m. And z plus y, of course, is the capital M, because they have to add up to the same thing. You can look at the ratio of mosquitoes to humans is m divided by h, as it should be. And number of bites per human per day is the same m into a, some quantity called a. Number of bites per human per day, is all of this can be sort of expanded out in this form, just to show you that these variables come in certain natural combinations. And once we think of this, we can come to the simplest model for malaria, and that's due to Ross and MacDonald. So the assumptions here is that the number of mosquitoes is constant. Call that V. Okay? Mosquitoes can be either susceptible or infectious. Call infectious mosquitoes I. Call the human population size to be capital H, as we did earlier. Humans can either be susceptible or they can be infectious, and call the infectious humans Y. Okay? Ignore incubation periods both within the human being and also within the mosquito. And remember that the rate at which mosquitoes bite humans is proportional to the number of mosquitoes, but independent of the number of people, because just mosquitoes are just going out and sort of looking for people, but typically there will be enough people to bite. So here is a model. So this is the same assumption that went in. This is the equation for the evolution of the number of infectious humans. Okay? So this increases as a function of the number of susceptible humans, which is h minus y divided by h, total number minus infected number divided by h. So this divided by h is a 1 over n factor that we were putting in when we discussed frequency dependent transmission. But this is also multiplied by mosquito biting rate, A, as well as mosquito to human transmission probability per bite, which is B. Again, this goes back to the same set of variables, our interpretation of beta that we actually put in, and how beta turned up in when we were thinking about the R naught factors. Exactly these two factors that turn up here, multiplied of course by I, which is the infectious mosquitoes. So notice that this is a little different from the earlier thing. Earlier I, S, etc. referred to one species. So now you're mixing infected mosquitoes with susceptible humans and asking that, all right, how does the infected number of humans change as a consequence of interacting with the mosquitoes? Then of course this term here is the recovery rate for humans, the rate over here, the psi here. You can write a similar term for the number of infected mosquitoes, which is here, which is the number of infected mosquitoes increases by a factor that depends upon this V minus I. So where was V? Number of mosquitoes minus number of infected mosquitoes. So that's susceptible mosquitoes times this factor Y, which is number of infected humans, scaled by the total number of humans here, times similar factors A and C. One is mosquito biting rate, and the other is human to mosquito transmission probability per bite. And again, this is the rate at which mosquitoes die, the minus delta I. So now that you are all very well experienced in SIR models, you can recognize exactly the logic that led to this. You should be able to write this out on your own. Now that I've described the problem to you and what different parts it has, this is something that should be possible for you to reconstruct. So from here, you can calculate a basic reproductive number, which I will not do, but I just want to motivate the different parts that exist to it. So this reproductive number is proportional to the product of two things. One is within the mosquito, one is for the human. So one is human and mosquito here that enters. Okay. And the reason we write it in this particular form is it has to involve these two parts. That is the rate at which mosquitoes get infected. There's a rate at which mosquitoes affect human beings, and human beings therefore get infected. So once you break it up into here and identify the factors that enter the A, B, the A, C, and the mosquito number of mosquitoes per person, you can immediately say it should take some form that looks like this. And here are the other factors. These are the factors, the gamma factors that turned up earlier. So that is DH and DM, the lifespan of the mosquito, and the duration of the lifetime of the human being. So they will turn up at this point here. So this is just the same as the calculation that we had done earlier, where we tried to find out what R0 was. And we said R0 had three parts to it. R0 was how long you were infected, what was the rate of being infected at that particular point due to your contacts, etc. So the contact term basically comes in the number of mosquitoes per person, and the A and B term go into how many of those are effective in transmitting the infection. Okay, so you can figure the logic out for that. And one thing that you can see from here is the ways to deal with this are you can either shorten the duration of infection in humans, thereby reducing DH. You can shorten the mosquito lifespan by reducing DM. You can reduce the number of mosquitoes altogether because those factors probably come in here, and reduce the biting rate, which is probably this factor B that enters at that particular point. Some things to note is that the well-mixed approximation doesn't work very well in this case. Mosquito behavior and biology are also important. Some people tend to get bitten much more by mosquitoes than by other people, than by other mosquitoes. Then incubation periods, multiple strains, all of these are important when we think about this whole problem in mosquitoes. So here is a little bit of history about Ross showed that the reduction of mosquito numbers below the transmission threshold countered malaria. Okay, and it's McDonald who showed that latency in the mosquito due to parasite development was also important when you thought about models. And what Ross and McDonald showed is that whether adult female mosquitoes survive or not is really the weakest element in the whole cycle of transmission. So that led to the World Health Organization deciding that let us use DDT in order to eliminate transmission, use DDT to kill mosquitoes, thereby eliminating that link in the transmission between mosquitoes and human beings. And of course, the latency of infection in humans is something we didn't mention, but that was also introduced by Anderson and Meba, who used an additional exposed class in human beings. So here's just a map of all these complicated models, which you don't have to look at, but I can just tell you. It's the earliest model due to Ross, which talked about susceptible, infected, and susceptible again in humans, with a susceptible infected population of mosquitoes. So this really is the sort of essentials of the model that I wrote down here. Then McDonald came along and basically looked at this model in a little more care, but eliminated the EM. This is the exposed mosquito. That is the part of the mosquito that has an internal life cycle. That was repaired by Anderson and May in 1991, who also introduced an EH class here, exposed for the human beings, to account for the life cycle within human beings. People looked at migration and visitation, the role of the environment, socioeconomic factors, immunities, immigration, etc., all of these, age structuring, which we talked about earlier, what is the role of different types of ages, what's the role of changes in population sizes, etc., etc. So all of the tricks, I'm just showing you this because all of the tricks you've already learned of how to incorporate age structuring, structured populations, migrations of populations, changes in population number due to deaths, all of those are ways in which you can generalize the simple model that we wrote down. Okay, so you should now be confident enough in your abilities to take a model like this and implement whatever you want. We say that, look, if I want to generalize it in this direction, I will be able to do that. Okay, so that's just a very quick introduction to a very large field, which is really the modeling of vector-borne diseases. So I hope you understood what I was trying to say. Let's now move on to the other parts that I wanted to tell you about, which is first is networks, and the second is individual-based or agent-based models. So network pictures you've probably seen a lot from Shatabra. I will probably redo some of what Shatabra did, maybe a good fraction of what Shatabra did, but it's probably good for you to listen to it once more. It'll just make you more confident in the material. This is, I think, a slide from a movie which shows a war, probably sort of large numbers of people moving from here to attack people on the other side. So another question is that when they made this movie, did they actually get real physical people to run across like this and run across in both these directions? If any of you have seen The Lord of the Rings, you'll see these huge battles with large numbers of people sort of attacking each other, the orcs attacking the dwarves and the elves and so on and so forth. So the answer is that the way these are simulated in a realistic manner is by using something called agent-based models. And that's, it's one place it turned up is very useful to think about these models. is precisely in animations and in making movies that really look very realistic, where you just couldn't imagine having 1 million people who you actually pay to come and run across the field. Certainly an expensive way of doing it. So let me talk about networks, but let me talk about networks in a slightly different way from which Shadabra would have talked about. And the sort of general idea is that you should think about different ways of looking at the same problem and becoming aware of different ways of formulating it. So this is a metro. I don't know which metro this is. Yeah, no, I think that's probably the London metro. This is Bombay. No, sorry, this is Chennai, likely over here. When you think of metros, you think of maps of metro stations. In the case, in the London metro is called the Tube. There are these very nice and complicated maps of these. And these look like, a section of that might look like this: Charing Cross, Cannon Street, Moorgate, etc., etc. If you spread these out in real physical space, depending upon where stations were, they would actually look like this. So something has to be done to take something like this and put it in this very nice geometrical form. Okay? And when the London Tube, the London Metro was first developed, people looked at pictures like this and they suddenly thought these distances are all very large, I don't feel like doing this, how do I go that far? And one way of encouraging people to use the tube was to draw a different map of the tube. They didn't so much focus on the distances between points as the connectivity between points. Okay, so what one person did, who was a person called Beck, who was a draftsman, he focused on the connections between stations, magnified the central region, brought in those from far away, laid the lines out so they ran either vertically or horizontally, or at worst at a 45-degree angle, and thereby compressed this information contained in this physical space into a space of connectivity. Just what is next to what, which station comes next to which station, and the fact that this may be 1 kilometer but this may be 20 kilometers doesn't matter at all. Okay? So if you want to be a little technical about it, he concentrated on the topological aspects of the network and not on its metric aspects that had to do with distances. So, as I said, he concentrated on the topological properties of the London Underground network. So that's actually what the full map looks like. It's a very complicated metro. So topological means that absolute distances don't matter, but how places are connected does. And that's the idea behind a network, mainly. So that's the Bombay train network. So social networks like that are networks modeling links between people. And of course, here distances don't make a difference at all. You could have the same link with someone in Bangalore as in Chennai, as in Delhi, or in New York. Any of these don't matter. What matters is the fact that you have a connection with a person. And once you have a connection, you draw a connectivity between them. Of course, you can have networks in many different variables. You can have emotional networks, transactional networks, where I sell something, you buy something, we have a transaction. There are some contact networks, we come close to each other, we are temporarily in contact. Could have sexual networks, and so on. So depending upon what it is that you want to study, you have a network that's adapted to that. A network of contacts is not the same as a network for emotional interactions. Here's one use of networks. So here's an example that says, once I have a flu case here, these are the people who were later infected as a consequence of that flu. So these are male students, female students, and probably household members are a member of this household. All members of the household are connected by this little circle here. These lines represent the transmission from one person to the other. And I think this is probably derived from a school. And people who were looking at swine flu at an elementary school in 2009 found the students were more likely to catch the flu from playmates of their own sex than from the students seated near them. So a boy was more likely to contact the flu from a boy, even though he may have been sitting next to a girl in class. Okay? So this is how the flu spread first through students as well as then later through their households here. Closing the school after the outbreaks, this is an interesting point, did not slow down the rate of transmission because friends kept meeting friends outside of school. It was really the contacts between people that were important in ensuring that this disease spread once it had started out. This is another sort of network depicting the spread of SARS in Taiwan in 2003. So totally 961 patients, 22 areas, 14 hospitals, 1,313 links. And you can see the role of the hospitals here, the hospital here, hospital here, hospital here. and hospitals to which a large number of patients went to when they actually fell ill. So every network is different. What you might hope to find is that social networks that are relevant to disease spread share some commonality that makes them special. So then you can ask what properties of a network determine how diseases spread upon that network, and what properties of the network determine how to control disease spread. Okay, so we're going to be looking at networks that are like this, and let's introduce some terminology. So, as I said, I'm going to repeat what Shritaba said, but I'm going to say it in my own words and probably slightly differently, so that it just helps people understand if they listen to something multiple times. So networks or graphs are made up of nodes or vertices, as well as edges or links between nodes or vertices. Okay, I can make any graph up using these two terms. Graphs are also called networks. We'll call them networks in this case. You can look at a network and say that, look, here is a group of sites that seems to be well connected within itself, but not so well connected to the outside. So this is a cluster of nodes over here with only one link to nodes outside that. Here is a particularly important node. It's called a hub because many things connect to it. Okay. Here is a triangular situation here, which is important for reasons that I will describe later to you. I can think of path from I to j as going from I to a particular site j in a particular way. Start at I, go here, go here, go here. That's the only way of going from I to j, although in principle, without sort of retracing your path. Although in principle, I guess you could go from here to here to here to here to here to here to here. But this is the shortest path between I and j. Okay. We can ask how do diseases spread on networks. So imagine that this is a network of contacts. We have one sick person inside there. These are the other persons that this infected person has contacted. So the way you do it is you start off with one node as an infected node. This node infects this node, and at this point 2 nodes have become infected. Now 3 nodes have become infected. Remember, you're only infecting those that are closest to you, that are at each time step. You're allowed, this person cannot be infected because this person has had no contact with this person directly. The contacts are only those that are marked by these links or edges between the different nodes. That's one more infection. But now this node has actually recovered. Okay? So I can look at this picture changing colors in time, things become red, things become green, etc. And that tells you how the disease evolves on the network. Some more terms. A hub, as I drew earlier, is a node with a number of links that greatly exceeds the average. For example, this node has 3 links, 1, 2, 3. This has 2 links, 1 and 2. This has 2 links, 1 and 2. This has 2 links, 1 and 2. This has just 1 link over there. But this node here has 1, 2, 3, 4, 5 links. So this is a hub. A hub is a node which has lots of links that emerge from it. When we think about disease spreading, these hubs that have a large, in quotes, degree, that is number of links that emanate from it, are important because they are really responsible for spreading the disease to lots of people. The technical term is a super spreader. Okay, so large degree nodes are potential super spreaders. We can talk about the path from I to j, which is some sequence of nodes connected by edges shown in blue, so 1 here, 1 here, and 1 here. A network is connected if a path exists for every pair of nodes. So choose one node here, choose a node there, and ask, is there a path? If the network is connected, you will always find a path. It can be a complicated path, but there will always be a path. A connected component of a network, you can have networks which you cannot go from here to here along a path. But a connected component is some subset where every pair is connected by a path, and you have no path that connects to nodes outside that little group there. Okay, so everything here is connected, no problem. But here is something that is disconnected from these. So all of this, I can go from any site here to any site here by just walking along the links. But I cannot go from here and here, between here and here, because there is nothing that connects. There is no link that connects this part of the network to that part. The order of the network is the number of nodes. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 over here. The size of the network is the number of edges. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, whatever it is. Some number that sits there. For a fully connected graph, I can determine the size of the network as n into n minus 1 divided by 2 because these are not directed. Okay. For a sparse graph, m, is much less than this number. So this is everything connected to everything else. Start with 1, attach it to n minus 1, divide by 2 because you don't want to overcount on that. The degree of the network is the average number of edges on a node, which is n times k divided by 2, thereby defining k. Okay. Distances on a network is the number of edges or links that you cross between I and j through the shortest path. So we emphasized the shortest path just now. And the characteristic path length of the network is some measure of the average length of the shortest path connecting every pair of vertices. So that's a natural definition. I don't want to talk about the shortest path between just this and this. I say look at every set of 2 nodes, find the shortest path between them, average that over all choices of 2 nodes, and then you have your average path length that comes in there. The smaller the average path length, the fewer the number of steps that you need to take to get anywhere on the network. So it's just in 2 steps or 3 steps you can get to any other node because everything is so well connected that it just doesn't. If you had a completely connected network, m equal to n into n minus 1 by 2, then in 1 step you can get anywhere because that's all it takes. Everything is connected to everything else. But in general, for a real network, the smaller it is, the fewer the number of steps that you need to take to get anywhere inside the network. The longest shortest path between any 2 nodes in a network is called the network diameter. Okay, that's another definition that you might want to use here. The giant component is if a single component contains the majority of nodes in the network, that's called a giant component. And here is a giant component where all of these are connected to each other. And the concept of the giant component is important when you think of disease propagation in networks, because usually whatever will limit the growth of the network is the size of the component itself. So what you want to ask is, does a network have a component that is as large as the network itself, or does it have a giant component, is important. Are we clear up till now exactly what we are doing? That we've just been talking about definitions so far. You've seen these definitions earlier, so you should be clear about what they mean. Now let's just drill in a little bit on other important quantities of a network that will affect how disease is spread on the network. Okay? So one is a measure that reflects the cohesion, and that is how do nodes talk to other nodes. So a node's degree counts how many edges it is connected to. So here's a link of degrees here. There's a map of the degrees. This is connected only to 1, so this has degree 1. This is connected to 3, 1, 2, 3, so it has degree 3. This is one of these really highly connected nodes because it's connected to 8, 1, 2, 3, 4, 5, 6, 7, 8. So given a network, I can write down the degrees that it has. And you can see that these degrees, each node has a different degree. There are some spread of degrees between the nodes. They don't have a different degree, a whole bunch that have 1, whole bunch that have 3, 2, etc., etc. But they're not the same for every node. Another word for node degree is also degree centrality, and it's used to determine how important nodes are in a network. And a simple way of determining is a node important or not in a network is simply to ask what is its degree. Okay? With me so far? Okay, so now let's just look at simple networks, and the simplest network is the regular network. So you may think, so this also looks like a network, but it's a particularly simple network to look at because it's arranged in such a nice regular manner. This is a 2-dimensional lattice, whereas this is a 1-dimensional lattice that can go only in one direction to get to a point here. If I want to go from here to here, I have only one variable that I can change, which is as I move along this. Whereas here, if I want to go between here and here, I can either go like this, I can go like this, I can go like this, to many other. So this is a 2-dimensional network, that's a 1-dimensional network. Here k is 4, each point is connected to 4 neighbors. Here k is 2 because each point is connected, so the degree is 2, and there the degree is 4 for everything. So regular networks have typically a large mean distance between nodes. If I wanted to get from here to here, I have to go to this. If I want to get, as I make it larger, if I want to go from here to here, I have to go here, here. And I can also do it in the other way. So here n is 17. So as I make n between n equal to 8 and n equal to 17, the distance between 2 nodes, in this case chosen to be opposite to each other, is as large as some number of order n. Let me take a regular network and then I can introduce what I call shortcuts. That is, just connections, pick any two nodes at random and put a connection between them. With shortcuts, is my job a little easier? The answer is yes. Because if I wanted to go from here to here, the best way to do it is to go from here to here, to go to here, shortcut directly, and then move here. Or I can move here to here to here to here to here, and then back there. That's certainly easier, and especially on a larger network, if I wanted to go from here to here, I would go here, 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 and then up to that side. So these shortcuts within the network, where you take a regular network, add a shortcut to it, you can get between points much faster. You reduce the mean distance between nodes. In this case, the probability or the degree distribution, the distribution of the k, is just a peak centered at one value. Okay? So here it's actually 4 for this, in 3 dimensions it is 6, but these are very simple degree distributions that you can have. So if you consider nodes exactly at the boundary, for example, this has 3, this one has only 2. So either you can say that, look, I'm going to put this all in the probability distribution, I'm going to have something here, I'm going to have a peak at 3, I'm going to have a peak at 2 in this particular case. Or I can say I'm going to take my network large enough so that all of the sites really are in the bulk and relatively less are in the boundary. We know that because suppose I think of this as a circle, the stuff inside the circle is pi r squared, but the stuff on the perimeter is just 2 pi r. Okay, so as I make the circle larger and larger, the r square will overpower the r. So most of the sites will be in the bulk and will not belong to the outside. So once I normalize this distribution, it's really only this particular term that counts and not those terms that come from the boundaries. But that's a good question. It's certainly, I'm not allowed to write down such a simple degree distribution because I have to account in the picture for the fact that it is more complicated. So here are your regular networks. Here are random networks. But this is not completely random for a reason that I will tell you now. It's not completely random because what it involves is what is called rewiring and creating shortcuts. That is, suppose this is my network of connections, here, 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 with this thing here, this thing here, this thing here. What I can do is remove, for example, this one and attach it to something else. So I don't change the number of links, I just rewire them. That's not the same as putting them in at random. Okay, that's important to stress. So I can rewire that to here by adding this term, so on and so forth. So it's not completely random because it's obtained by a very specific process. When we discuss randomness in any mathematical context or any computational context, we have to specify what we mean by that. Random in one context may not be random in another context. And the reason I sort of showed you that is basically to contrast it with the case where I take two nodes at random and I put a link between them with some probability p. So I connect each pair of nodes with probability p, and if I gave you some set of nodes connected in this manner, what is the degree distribution that it would have? So we know the degree distribution in this particularly simple case over here. Each thing, assuming that I'm only going to talk about the bulk, each has only 4 neighbors, so that's a peak here. If I did this in 3 dimensions, it would have a peak here at 6, because it has 6 neighbors. But suppose I did this particular algorithm of just picking 2 things, adding probability p, picking 2 things, adding a probability p, adding with probability p. So then I get some complicated mess that looks like this. What is the degree distribution of that mess? Okay? So the answer here is that that is a Poisson distribution. Here is a fit to the Poisson distribution, so a very large number, 10 to the 6. And this is a random graph with an average value of degree that peaks at 3. Okay, and this of course depends on the value of p. If p equal to 1, I just have everything connected to everything else sooner or later. But this is some value of p that says roughly a fraction p of links are there and 1 minus p are not there at all, and they are all connected at random. So here are the limits, the regular network which had one sharp peak at one value, and here is the Poisson distribution that has a slightly distorted peak here at some value here. So now let's ask the question, let me go back to thinking about diseases. in a model for diseases where I know the people who, the children in school, the boys and girls talking to each other, the SARS, the hospital, etc. What is the network that describes how they interact with each other? What is the real network that underlies diseases? So before we get there, we have to talk about a few more properties, and then I will tell you what makes those networks special or interesting. And again, repeating what Shitab has already done. And that's the property of transitivity and clustering. So, clustering refers to the fact that friends of friends are more likely to be friends of each other. If I am your friend and you are her friend, and she is more likely to be her friend rather than sort of completely somebody that I don't know at all. That's just the structure of how friendships work. Okay? So transitivity in the network context means that if you take persons A, B, and C, or node A, B, and C, there will be an edge connecting AB, AC, and BC. And this refers to the tendency to form triangles between nodes. And you measure this quantitatively through a clustering coefficient. So this is transitive because 1 to 2, 2 to 3, 3 back to 1. This is intransitive because 3 is a friend of 1, 2 is a friend of 3, but 1 and 2 are not friends at all. So roughly speaking, the existence of triangles of this form in a network is a measure of how transitive the network is. So this sort of analysis reveals tightly connected groups or communities, and you calculate the clustering as the ratio between the observed number of closed triplets and the maximum number of closed triplets that is possible in the graph. And clustering and transitivity have similar definitions. So how does all of this apply to the networks that we're interested in when we think about disease? The answer is that many biological and social networks have topologies that lie between either being completely regular or between being completely random. And this is an idea that Watson-Strogatz introduced in 1998. And that goes to the idea of small world networks, which you've probably heard about in detail from Shitabro. Did Shitabro tell you about the Milgram experiment? So you know about that. So it's this Milgram set volunteers the task of sending a letter to someone completely unknown to them. He just said, send it to somebody you do know who might be closer to them, and ask them to send it on. Okay? Then after that, once they arrived at the recipients, Milgram could see how many steps this letter had taken to reach that person. And it turned out what was surprising was that the number of steps was typically not very large. If I don't know somebody in, Begusarai, for example, and I'm given the address of that person, I have to try to figure out how to get my letter to that person in Begusarai. If I normally thought, look, there's no chance that I have of knowing that person, knowing anybody that that person knows, I might have thought that the route that my letter would take would be a very long and complicated route. However, it turns out, interestingly, that for human interaction, that number is actually fairly small. And this is popularized as something called the small world phenomenon, or the phenomenon of 6 degrees of separation, because typically what Milgram found was that there were 6 intermediate steps between persons that it took to get from here to there. The small world network is obtained by taking a regular lattice and then just adding connections that are long-range connections at random. The same thing that we did when we say with probability P, attach link 1 and link 2. Any link I, any link j. So it's interesting that small world topological networks seem to look a little bit like regular lattices. If you sort of look at them, they have more or less the type of lattice structure that we wanted. But they also have these long links that are also a feature of the random network. The random network doesn't care. I pick something here, pick something there and connect. It doesn't care whether it's in the other end of the world or it doesn't care whether it's in the same room as you. So this is something in between the lattice and the completely random system. And what's interesting is that, like the lattice networks, their clustering coefficient is high, okay, compared to small world networks, but their path length is small, like a random graph. It's very easy to get from one point to another point in a completely random graph that remains connected, although the neighborhood of this random graph looks nothing like the neighborhood of a lattice, because their connectivity is very different, the clustering is very different. So here is a regular network and made in a slightly complicated way. Here is a small world network that we have. This is the path length as a function of the rewiring probability. So remember how we rewired. We took an existing connection that was there and we just linked it to something else, taking care to see that the network didn't split up altogether. Because the moment I split a network up, there's no way of getting from here to here once I have disconnected components. So the secret is to randomize the network without introducing disconnected components into it. And you can see that there are two quantities here. One is relative path length and relative transitivity. So transitivity stays high, path length goes down very sharply as you increase the rewiring probability. So real networks appear to be somewhere in between regular lattices and random networks. So here is some example of I forget what these quantities are labeled as, but I think this should be average path length which comes down very fast, and transitivity that comes here. So these have high transitivity and relatively low path length. Okay. So here are actual numbers from a review article by Baradbasi and Albert, which shows you what the average path length is. So this is math co-authorship, substrate. These are system biology graphs of connections between protein molecules here, food webs. These are ecological examples here, power grids, C. elegans neural networks, movie actors, etc., etc., etc. And here is their clustering coefficient. So this must be the path length here that is identified here for these large networks. Path lengths tend to be small. Clustering coefficients tend to be high compared to those for random networks. If you took the same size of network, just connected things at randomly, this would be the randomized cluster coefficient there. And this is the real cluster coefficient. So look at the difference between this. So 0.76 and 5.5 to 10 power minus 5. So these networks remain highly clustered even though they have a short path length. And that is a crucial idea. It's a very clever idea that Watts and Strogatz actually found out when they began to discuss the phenomenon of small world networks. So now the question is that all that there is? I sort of made a case for you that most natural networks, social networks, etc., had a small world property. And for a brief while, this was the idea that dominated thinking about sort of diseases, social networks, etc. But it turns out that real networks seem to have somewhat more complicated problems that are not completely captured in this description. Okay? And that is the nature of the degree distribution. So first of all, here it says most social, biological, technological networks have patterns of connection between the elements that are not either purely regular or purely random. And we saw an example of that. Short path lengths and high transitivity was our example. But other features include the heavy tail, a long tail in the degree distribution, high clustering, which we discussed, community structure, hierarchical structure, etc., which Shitabro has discussed in some detail. So this is the degree distribution of a random network. As I said, it's a Poisson distribution. This is what real networks tend to look like. Okay? So the Poisson distribution comes up like this, has a peak and comes down. These tend to be sort of fairly highly linked. So low linked here. So there's relatively small probability that you will have highly linked nodes here. Relatively small probability to have low linked nodes here. Typically most of them center around some average that sits here, and that's the peak. Okay? Contrast that with this. Most nodes have fairly low links, 1, 2, 3, etc. But some nodes have a very high amount of linkage. They're way off on the right hand side compared to that. And this is, Shitabro must have introduced the term the power law. There's a power law tail to this, a heavy tail to this distribution, suggesting that there is an importance to those nodes that are very, very highly linked to many other types of nodes. Okay? So that's what real data looks like. And we went between this degree distribution for regular networks, this degree distribution for random networks, and this degree distribution for what I call scale-free networks, which is that example that I gave, where the degree distribution decayed as a power law. Okay? So again, come back to the question, what do networks relevant to disease spread look like? How do we find them? What properties characterize them? And what does network science tell us about how to stop disease propagating across the population? So both scale-free and small world networks have specific structural features. Here you have power law degree distribution for the former and short path lengths and high clustering for the latter. Regular networks have long average distances between nodes and high clustering. Random networks have small average distance between nodes and small clustering. Small world networks are in between these. And scale-free networks have a complex degree distribution where you have some nodes that are very, very, very well connected. So here are real data pieces. This just shows you the networks of contacts between intravenous drug users. So these are the people who are distributing the drugs, so they tend to be particularly well connected. And these circles are the users of the drugs. These are movement of cattle. Remember we talked about the foot and mouth disease where we had to be conscious of where cattle were moving between markets. This is sort of cattle movements in Great Britain from 6 farms, color-coded by the farm. This is the network of contacts between individuals in a geriatric hospital. So you have all of these. So these are people who are, so the size of these circles is the number of contacts, is the degree of each other formed by this network over this time. This is a nice example where you can give wireless contacts to all students in a school. And the moment one student comes close to another student, the two wireless contacts are linked to each other. So this one records the presence of another student on that. And it can maintain this record and upload it to the web. And later you can use it to construct exactly a network of this form. So you actually know who contacted who, when they contacted, what was the period of contact. You can neglect minor contacts, a couple of seconds of somebody just walking past each other. But you can keep contacts that are more than 5 minutes or 3 minutes, suggesting there was some social interaction between them at that particular point. What's known about epidemics on small world networks is if you take an SIR model over here, infected individuals, blah, 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 you removal, etc. The critical infectiousness at which the disease infects half the population decreases with P. I can ask the same questions, take an SIR model, put it on a network with a specified P, a small world network with a specified P, and see, all right, typically how did the disease spread? How long did it take for it to affect half the population? And using just a single quantity R, R half, which I can vary, that's just the strength of the rate at which I can infect somebody, you find that as I increase the P, the R half decreases. Okay? And typically, only a relatively minor rewiring means that the disease basically spreads as fast as it would have on a random network. And that's interesting. It says that even though you've gone to this great trouble of being very close to the regular lattice, just introducing a very small 1%, 5% number of these very long range links ensures that the infection will go all over the place. Okay? So that's something else that is a bit surprising. There are various measures of number of infections on different types of graph, an Erdős-Renyi random graph, a fixed degree graph, a scale-free graph, and that's what it looks like. This is the scale-free version, this is the Erdős-Renyi curve, etc., etc. So the nature of the network influences the dynamics of the disease on that network. That's essentially what this graph shows you. Diseases spread more rapidly on small world networks than on regular ones. It's more difficult to control, but it's important here to look at these shortcuts, the people who connect one node here to a very large node here. So the classical example of this is truck drivers for the AIDS epidemic. Truck drivers are people who crisscross the country. When they stop at the various stops, typically they mean, often not typically, sometimes they mean sex workers in that region. So the spread of AIDS in a country typically is connected to the people who move these large distances while belonging to a high-risk population. So they're called super spreaders. The time it takes for an infection to propagate on a highly connected network can be very short, basically the same as a random network. For scale-free network, there is actually no threshold. Pretty much any value of R, probability of spreading the disease to a neighbor, to someone you're connected to on the network, will ensure that it spreads throughout. So that's an interesting result. If real human networks are scale-free networks, then you have no hope of limiting it because R0 is effectively infinity. There is no threshold value of R0 which you can control and try to bring down. So that's one interesting negative result there. I'll just spend a few more minutes talking about epidemics in a globalized world. So we talked about one set of networks here, but it's a little more complicated when it comes to real life. And real life is complicated because when you think about epidemic diseases or pandemic diseases, remember I said epidemic diseases are maybe localized to one country. Pandemic is something that covers many countries. And H1N1 was classified as a pandemic because many countries were affected by H1N1 influenza. Okay? So here you can look at an S plus I going to 2I going to 0 sort of model, and of basically it's a sort of SI model, SIR model, which you can have infections, people recovering with some beta here. And this is used to look at SARS with local infection dynamics, but also allowing people to travel. So one reason SARS spread so fast started off in the Far East, in Hong Kong, I think, and from China and then moved to Hong Kong. And then there were cases reported in Toronto and then later in the US. That happened because somebody with the disease took a long distance flight that moved from Hong Kong to Toronto. So what these people say, what this paper and later papers say, is that in our modern world, the fact that people can fly long distances really aids the transmission of a pandemic. Okay, it elevates what could have been a purely local epidemic 100 years ago, 150 years ago, to something that can spread across the world. In order to do that, you need to know all of these connections. For example, the flights that come here to here, here to here, here to here, here to here. So what these people did was to look at all the airports among the top 500 largest airports by capacity. So they constructed a global aviation network, and then they divided the population into local populations labeled I, containing NI individuals, and used that as a starting point for a stochastic SIR model. So how people moved was determined by some matrix of transition probabilities among populations. So you could actually move both locally as well as long distance through air. You can weight links representing flights by passenger traffic through the relevant airports. So very busy airports had lots of links coming out of it, lots of flights coming in and out. And relatively small airports had fewer links coming out of it. So you can say that that much more traffic is seen by an airport that has many people flying in and out. So with this, they were able to predict the number of cases changing in time. So this is a geographical representation of SARS cases on 2003 as reported by WHO. First happened in China. Disease was then carried to Hong Kong on February 21st, 2003, and then began spreading through world aviation routes. This is the simulation result. So simulations 90 days after initial infection in Hong Kong corresponds to real SARS infection at the end of May 2003. And that's how you sort of see the relationship between what they predict here for the number of cases. And they have a nice graph in their sort of table in their paper, which says here are the simulation results for the maximum and minimum number of cases. Here is some average, and here is the WHO measure for the number of cases in Hong Kong, Taiwan, Singapore, Japan, Canada, etc. And here is the relationship between the average and the number of cases predicted by the WHO. And you can see that, you can more or less there is some correlation between this. Okay, so these methods really tell you how to take complicated network information that you might not even have thought of. It was hard to think of earlier as air travel being really the predominant way in which you took diseased people from one place to another place very, very far away and allowed them to spread their disease. So this is a paper called Role of Airline Transportation Network in Prediction and Predictability, and this particular group now has a sort of large computer program that is able to run moment there's any disease spread to say what happens if we factor this global airline transportation network into the model. Okay, so let me take a break here. When I come back, I'll talk a little bit about agent-based models. Then I'll describe the tutorial to you. And basically the tutorial, I want you to do it on your own. So you can either sit here or just hang around briefly. But this is something that you can try out on your own. So let's take a break now if the coffee has come, or you can ask questions. Since we did both vectors, we did a complicated set of things today. We did vectors, we talked about networks, we did a whole bunch of different things. Are there any questions that you have? So the advantage is that a priori you don't know what social networks look like. Either you must get detailed social network information from basically doing a census of your population to figure out who knows who, or you can try and say, can I think of a network that has the sort of properties of a small world network, that has the properties that I think might be relevant to how people interact with each other. So I know from the small world experiment that it actually doesn't take too many steps to go from one person to the other. So even though they look completely disconnected. So even though also I know that typically people have a well-defined neighborhood of people that is local, that they know. So one way of doing it, a very clever way of doing it, is this idea of a small world network. You start with a perfectly regular arrangement, a regular lattice, and then you rewire to introduce these long-range connections. So it's more trying to incorporate what was known from Milgram's initial experiment. We just came up with the fact that typically any two people, you only need 6 people in between at max to connect them. So the idea was to take that and convert it into something like a model network, which is what the small world network does. But the difference is that later it seemed that that was more complicated than it seemed, that you did need to have this very broad distribution of degrees, very highly connected people and a large number of people with low levels of connection. And that might be again a sort of new twist. All of this, in principle, you can do it in two ways. Either you can find out in detail social networks for everybody, or you can say, let me take the little information that I have about these networks and try and make a model network that simulates some part of that. So that's the sense in which the small world network was actually introduced. It's an assumption that encodes this fact that you have a relatively small distance between any two nodes in the network. You can show that the distance grows only logarithmically with the size of the network. If the human population is about a billion, a few billion, so it's 10 to the 9. The log of that is around 9 people. So which is why the 6 degrees, 7 degrees of separation actually comes in. That's because it only grows as a log of the size of the population. So that's it. So usually when there is a disease, people try everything on it. So people try random networks, they try small world networks, they try Erdős-Rényi, they try these sort of long distribution, the long tails of the distribution networks. But certainly a small world network would be an interesting thing to try right in the beginning, because that does have some known properties of real networks. So topologically in the sense that it topologically encodes the fact that you have locally a well-defined neighborhood, but also the small fraction of long-distance links. So that topological feature which we believe is responsible for small world behavior is encoded in the construction of the small world network. No more questions?
You talked about headings for us, so that is noise over here, but like yesterday. So when do I use multiplicative noise and when do you use additive noise?
So they turn up in different contexts, and I think the example that I gave you yesterday was additive noise actually, because the way I wrote it out was actually additive. It didn't depend upon the S. But if I had a term that was proportional to the number of S's or the number of I's, that would, if I had a noise term that multiplied S or I, then that would be multiplicative. Usually multiplicative noise problems are a little more complicated to deal with, and additive noise problems are simpler. So often you might want to go to a limit where you look at additive noise. But again, it depends upon what is the problem that you want to look at.
Yeah, so like in what context of biology do I use multiplicative?
So one example might be that if you look at the term, the di by dt term, which is beta times I s divided by n, I can make beta have some value beta naught plus some noise term zeta here. And this term could be a function of t. So this is because beta represents the transmission of the disease between individuals. This might represent the effects of day-to-day changes in weather or climate that make it a little more or a little less possible that I might transfer the disease to you. This now will multiply the combination I times S, so it's multiplicative in that sense. Okay, but I could perfectly well add, all right, some other term here that depended, put the term here and then added a I here. That would again be multiplied. I could just add a zeta naught just there. This would just say there's some fluctuation in the number of i's. I don't know where it comes from. It's just added to the system. Maybe there's some outside perturbation that changes the value of I in some random manner that I cannot predict. So this would be additive noise because it doesn't multiply any of the basic variables inside here. Even if I multiplied this by s, that would be additive as far as this equation was concerned because it doesn't involve the I. So it depends on what you try to model, and in some situations the additive noise approximation is right. In some situations the multiplicative noise approximation is correct. It depends upon where you think is the source of stochasticity and how big it is. Discuss agent-based models fairly quickly, and then I'll go on to show you what the tutorial is and what I expect you to do. So agent-based models are sometimes called individual-based models, and you can think of it as the network that we drew earlier. Imagine that just instead of having boring nodes on those networks, you actually account for the fact that these are different people. That's one thing that you can do. And by the fact that these are different people, men, women, dressed differently, etc., you can put more complication into it than there is in the simple network picture. You can say maybe this is a highly susceptible person, this is a highly non-susceptible person, these persons contact each other less or more, and these contacts also evolve between the day and the night, for example. So this might be an office situation because everybody is dressed for office, but you might imagine at the end of the day they all go back home and then their contacts are with a completely different set of people. That's very hard to accommodate in a conventional network situation because that would mean a network that is time dependent in a particular way. But not only is it time dependent, it's also stochastic because you might go home, but somebody in your family may have gone out. So today you might meet them, tomorrow you might not meet them. So a dynamical network with stochasticity might be the best way to adapt these network ideas into this bit of the real complication of real life. Of course, real life is even more complicated than that because people are sort of constantly interacting with each other and moving along. The dynamics of how people move between different situations, etc., is itself very interesting on its own right. So here is an example of real individuals, a picture of real individuals. And the interesting thing to note about this is it doesn't have a relation to what I'm going to tell you, but look at how people figure out how to avoid each other, even though they're walking in perpendicular directions. So we anticipate how other people are going to move and sort of decide our own motion accordingly. We slow down, we speed up in order to account for that. So these models are used very much in models for transportation, economic needs, etc., etc., and also a little more in models of the type that I will describe increasingly. What we need to do in order to map any real system into an agent-based model is to figure out how the entities that are starting out, the people over here, map onto what we call agents. What are the properties of these entities that we want to abstract in the form of agents? And how do we take the interactions between these entities and represent them as interaction between agents? Okay? So the idea behind agent-based model is that you want to treat individuals in time and evolving in time and use that to make predictions about how diseases might spread. So as I said, this is an example of how you can simulate very large groups of people for a movie using agent-based methods. So this takes is actually very fast on PC with certain specialized softwares. Agents in a multi-agent system have several important characteristics. That is, each of these individuals are independent of each other. So they're called autonomous. The second is that the agent makes a decision to move, not to move, go here, fall infected, etc., based on its local neighborhood. So the agent doesn't see the full system, but only the neighborhood. Some of these will become clear as I move to examples after this, but just keep these ideas in mind. And the third is decentralization. There is nothing that tells agents what to do. Each agent makes their own decision about how to move, when to move, what interactions to have. So you can think of these agents as idealizations really of individuals with some decision-making capacity or able to change their own behavior based on their changing input that is confined to a local environment. So one example that I want to show you is of agent-based modeling is an example of cholera, which is, of course, is mainly eliminated in regions that can provide clean water, adequate hygiene and sanitation, but it still remains a threat in many parts of Africa and Asia. So here is a model, which is an agent-based model for how cholera might spread or spread in real terms in a refugee camp in Kenya near the border, where both sanitation was bad and housing conditions contributed to cholera outbreaks. So that's the paper that by a person called Andrew Crooks, etc. And this is the interaction between an individual, a particular agent, the host, the environment, and rainfall. So the way this works with cholera is that the host can go between susceptible, exposed, infected, etc., but in the infected stage, they can excrete feces which contain the cholera bacterium. This, using either runoff or seepage, can move into the water that is ingested by a susceptible host. The amount of water available to take this and convert this into runoff or seepage depends upon the rainfall. So that's an external or exogenous environmental variable. This is what happens to each individual here. Some individuals can die, some can recover, etc. So this is an SEIR model with two infected states. This is the interaction with the environment, both in terms of rainfall, as well as in terms of the nature of the water, where the water itself can carry the infection or not. Okay? So one element of this is certainly hydrology, which is the pattern by which water flows from point to point. The agents are refugees in the camp. And as the paper describes, the main agent in the model is a refugee agent who represents an individual refugee in the particular refugee camp. A refugee agent has a family and a home location. Agents of the family cooperate and share resources, but agents are instantiated, that is created with different attributes that contribute to why they are heterogeneous. So the whole idea behind agent-based modeling is to make some account or to incorporate some of the heterogeneity between different individuals in a population. So for example, you can have both male and female agents, you can have differences according to sex, you can have differences according to social ties, you can have symptomatic or asymptomatic individuals based on their resistance to being infected by cholera. Agents are mobile and purpose oriented. So for example, you can prescribe a set of goals for an agent, that is at 9 o'clock in the morning go from here and collect water from there. 3 in the afternoon go from here and go and buy groceries somewhere else. Go to school at 5 in the morning or whatever it is. So there are both activities that are accounted for. So location of residence, school, water point, religious center, market, food, all of these are assigned when you set up the simulation. And then these agents move back and forth. Depending upon the agent, their behavior is then prescribed. The problem with these models is that they're complicated. There are many variables to worry about. How many agents do you start out with? So this particular simulation had between 50,000 and 500,000, which is kind of reasonable for the refugee camp as it turns out. It looked at the daily water consumption, dehydration rate, symptomatic to asymptomatic, rate of return susceptible, distance from home to open field latrine from which there was runoff. All of these were prescribed, and usually the difficulty with these models is trying to understand this large number of parameters that has to be specified before you can actually do anything with it. So these are the three refugee camps, Dagahale, Ifo and Hagadera. And over here, this is where the camp is actually located. And you can see this is a map that is built up using a geographical information system that locates the mosque, the distribution center, the schools, etc., etc. So if you have access to a good, what is called a GIS, where all of these locations are marked, you know where these places are. Okay. Here is a sort of map of numbers of infected people as a function of time in these three different camps. So it's over a short time, 1 to 90 days, 90 to 180 days, to 181 to 70 days. And at this point, you can see how the number of infected people between 0, 4 to 7, 1925, 1 to 3, etc. changes by looking at the color coding on this. On this map is also marked clean water points as well as the camps. So these grey areas are the camps where people stay, each individual house. You can do different types of approaches here. For example, you can, instead of taking real details from the real population that you want to study, you can make a synthetic population that has the same statistical property. I'll tell you a little bit about this later. You can use census and demographic data and then assign a contact network based on how people interact with each other. And this is the idea behind one very famous agent-based modeling technique called Epidemics. Okay, so you can see how it's complicated. You have to deal with individuals. You have to deal with how individuals interact in terms of a network that is dynamical. You have to put it on some spatial background over here. And finally, all of this comes up with the sort of answers that how do the number of infected and where they stay change as a function of time. So this is probably a good point to tell you a little bit about our own work in this area and why we are interested in it. And that's our own work is really thinking about agent-based models for disease spread in real populations. And one set of diseases, the diseases that spread to direct contact. That is, if you come close to me, there's some probability that you will pick up the disease from me. But what we're more interested in right now, what we're thinking about, is diseases spreading through vectors. And this is important because the real major public health challenges right now are diseases like dengue, chikungunya, influenza, etc., etc. And dengue, chikungunya, malaria are all of them vector-borne diseases. And that's why we need to concentrate upon this. And the second part of it is thinking about how to develop good synthetic population models for disease studies. So the whole idea is think about a bunch of individuals, and normally you would classify them according to status with respect to disease, so either susceptible, infected, etc., etc. But now instead of a compartment, I keep track of all of these as individuals. Okay. There are other inputs that go into it. For example, male, female, age distribution, the state of their health susceptibility, what socioeconomic class they come from, and where do they stay. All of this we can actually load into the model. So just to imagine it, think that I have a grid here. Each of these circles is an agent in my definition. Okay? Agents, these orange agents are susceptible agents, but these are infected agents. So the rule is that these agents can move around. For example, here is a road. The agent can move along here to here. Suppose office is here and home is here. The agent can choose to move from here to here. Or the school is there. The agent can move along roads to the school. Agents, of course, have complex social behavior. They work in buildings, they go to markets, they visit theaters, they go to stadiums, etc., etc. But the fundamental rule of infection is a distance-based rule in this particular case. That provided I have an infected agent here, within some distance of that infected agent, if there are susceptible agents, you can transfer the infection to the susceptible agent. It's exactly like the compartmental model, except I now think of this in space. I can think of each of you in this room as an individual agent. If I am sick and I come close to you, within a certain radius, you have some chance of picking up the disease from me. Okay. If these are diseases that are spread through direct contact. So agents transfer infections to other agents, they can recover, and agent motion follows a global clock. What does that mean? That means that pretty much all of us between the time of 9 and 10 are going to college or going to university or going to school or going to work, and by about 5 or 6 in the evening we're all coming back. So there's this regular cycle which may differ slightly between individuals, but more or less everybody looks at the clock and decides what it is that they have to do. Okay. So here's one example of a simulation. So this is from the local area here. It's an area called Saidapet. So this is the Adayar river here. These are the bridges that go along, and these are all sort of locations. So this is taken from a map, what's called OpenStreetMap for this area of Chennai. All of these are people, are simulations of people, and these people go to work and come back. So there's a 12-hour cycle by which they go to work and they come back home. So suddenly you'll see them moving all together. They're all gone to work. You assign a workplace and a home. They dash there and they dash back. Okay, so that's kind of what we have in mind when we think of these models. You can take these models and run them. So let me just run this a little faster. Maybe I can get here at this point. You can see these red people here are people who have gone to hospitals. So this is the location of a hospital. There was one more hospital somewhere here. So these people, by the same rules that I had, an agent comes close to another agent that is infected, falls ill, they manage to go to hospital. Once they're in hospital, they stay in the hospital until they recover. Okay? The recovered agents are these blue agents, I think. Maybe it's this color here. And this evolves as you go forward in time. You go later and later, the nature of the colors change in this particular case. I can take that, and in my simulation, say for 1,000 agents, I can say, okay, let me run my simulation multiple times, and let me see what the number of susceptible, the number of infected, the number of recovered is actually doing. Okay, so I can generate curves from here because this is now stochastic. When two people come together, it's not 100% assured they will transfer the infection. There is some probability of transferring the infection, and this is done through the same sort of Monte Carlo method that I described to you earlier. So this is standard sort of discrete time Monte Carlo, but one could also do the Gillespie methods that I described to you, at least in principle. You know the simple Gillespie method, just imagine that it's complicated by a factor of about 10 million or so in order to do this. Note that these are, as I said, in each run these are different, but on average they correspond to the sort of SIR behavior that you see here. Okay, this has to be much more complicated, of course. We have to add home, schools, workplaces, playgrounds, occupation, social interaction, injury susceptibility, age, details of infection, etc., etc., etc. But finally, the aim is to study stochastic agent-based models for how diseases are transmitted between people. Unfortunately, the sort of data that you might need is just often not available at all. You might be able to figure out what home, schools, workplaces, and playgrounds are just by asking around. But what occupations do people have? Hard to figure out until you actually ask them. What are their social interactions and networks? It's hard to figure out who knows unless again you sort of record this information by going to each individual. These are a little easier to do. The details of the infection are of course a matter of basic biology. So that at least biology and clinical studies can tell you about that. More complex and realistic geographies can be put in. And vector-borne diseases, of course, is another question because that involves both the dynamics of the vector, which is not known, as well as the dynamics of the human beings who are bitten by the vector. So we've been working with clinicians at this particular very famous hospital called the Christian Medical College Hospital in Vellore. So Vellore is about 120 and 30 kilometers from here, pretty much on the line that joins Chennai and Bangalore. So it's about an hour and a half to 2 hours outside of Chennai. It's a large hospital, and what this group has been doing is running population-based censuses of a large population in and around Vellore. So this is actually two populations. One can be called an urban slum, and the other is sort of village areas around it. So about 40,000 in the urban area and about 120,000 individuals outside. Okay, so they know many details about these people. It's a fairly regular census. It happens every year. So these are the 4 units together. Population of about 40,000. They know the religious composition. They know what people work at, And they also know that most of these people are daily wage earners. Okay, what do they get? Information that they get is where do people stay by latitude and longitude, how many members are there in the family, how many adults are there, how many children are there, what is the level of education of each of the family members, their occupation, sex, year of birth and marital status, number of rooms, is the house owned or rented, does the house have a vehicle, a phone, a fridge, a washing machine, etc. You might wonder why they're asking these questions. The answer is that often you can use these as a proxy for socio-economic class. And then you can ask, does disease tend to affect people who are, depending upon their socio-economic class, are they a little more or a little less protected against a particular disease? Often this is a function of many, many variables, because you might imagine that with a better socio-economic class, levels of sanitation that they encounter might be better. But again, it's interesting to ask, where do all of these variables actually fit in? And how do they influence the progression of either of an infectious disease or even a non-infectious disease? You might want to ask, for example, against the question that we study here, that if you have low birth weight of infants, is a big problem in India. But what does it correlate to? Does it correlate to levels of education? Does it correlate to levels of diet? Does it correlate to the nature of the family, to socioeconomic class? Does it correlate to air pollution? Does it correlate to water pollution? These are questions that we'd like to figure out, because to actually tackle this problem, you have a lot of data, but to try and get useful information out of that data is always very difficult. So what we've taken is a small part of the census data from this population a few years ago. And we know there are 522 individuals of age less than 60 and 344 of age greater than 60, so many of age in 60. And then we have to make certain assumptions. The assumptions we make is that those of age less than 16 are assigned to one of two schools at random. These are individuals who stay at home. Half of these go to work, half of them stay at home. We put in workplaces at random and assign an agent of workplace in the beginning. Here is the age distribution. Here is the number of residents per household. It's a large household with 11 people, but typically the peak is at around 4. You can read this data directly from a file. These individuals are assigned a household ID in the CMC data. We use an SCIR model with 3 levels of sickness, mild, moderate, and severe. There's one hospital and agents go to that hospital. We start these, as with all of these simulations, with one infected person, and then we ask, how does it spread? And we can change the person that is infected, and you say the infection is equally transmitted with equal probability between all agents who share the home or the workplace, but also at the hospital, where this person encounters doctors and nurses and so on. And we could account also for public transport, but we don't do that. Okay, so here's a sort of tiny example of how you can have people here going to work, coming back. So it's a sort of small family that sits here. Somebody goes to school, somebody goes to work, or workplace 1, workplace 2, and then comes back. And then you can make this more complicated and look at the case of, for example, slow infection. So this is at night, this is going to work, this is coming back, this is going to work. I think these things are probably hospitals over here. And at some point, the color will change as people get infected. And then you can sort of, they tend to hang around in the hospital here. It's a little more cleanly seen if you look at the case of a fast infection. And sorry for the light here, but here it's an infection with a larger value of R0, which propagates faster among people. And if I just speed this up a little bit, you might be able to see here, the color has changed. It's not a good picture, this, unfortunately. Here's a much more complicated picture of 22,000 agents. And here, this is an area of Chennai, which includes Egmore, Nungamak, Roypada, Chepauk, and Centra. It's an SIR model, so green is susceptible, R is infected, blue is recovered. And then I can sort of do the same thing. You can see people dashing back and forth. And this is a fast infection, so there's a whole bunch of red people that congregate suddenly here. Okay, because those get infected. This is actually the Vellore data, done with 70 agents, of which 70 are infected. I think 70 are infected, about 1,000 odd agents. This is actually all of that different. We know exactly where people stay, so we can put that in. We can take one infected agent, we can identify the hospitals and have people move backward and forward between them as they fall in. You can see over here how the S and the I and the R actually behave. And now, because it's a stochastic model, you can see that sometimes it doesn't change, sometimes it goes, sometimes it changes. So these should all be smooth curves if you are according to the SIR. But in this case, you can see the discreteness as one person falls sick, one more person falls sick, etc., etc., etc. The advantage with things like this is that you can ask specific questions. You can say people go to hospital when they're ill. How many people go to hospital and how much does that number change? So these are sort of 3 runs where you look at the hospitalized individuals. It starts here, the epidemic takes off here, it goes, it peaks, and then it comes down. The standard behavior of I as a function of T. Except now all of these individuals are going to one particular hospital in their neighborhood. The advantage here is you can say that at the peak of this infection, the number of people in hospital is about 90. Now the question is, can your hospital hold 90 people? Maybe the hospital has only 25 beds or 30 beds. In which case, if you want to prepare for an epidemic, you must say, I must make arrangements for the hospital to have 60 additional beds. And that is going to be part of my planning for a particular epidemic. So if I suspect that an epidemic is going to hit a particular locality or a particular region, I can run these simulations and ask, what are the numbers that I expect reporting to this particular hospital if all of the people in this area potentially report to that hospital? And that will depend upon the details of the disease, because the R naught will determine how many people get infected, what are the peak values. It will depend upon where it started from. And it could also change depending upon what I put in to control the disease. Suppose I said, okay, there is a disease, there's already Chennai has got various cases of Ebola have been reported, or Nipah or something. All schools are shut down in the following districts for the next 1 week until we figure out what's going on. That has now seriously changed the patterns by which people interact or people mix. And in these models, because these models are powerful enough, I can ask that question. I can just say, all right, everybody in the age group of 3 years to 16 years stops going to school from tomorrow onwards. And then I can say, what difference does that make to a curve like this? Do I expect it to rise at high, or do I expect it only to rise here? And because these are stochastic models, I can check out different things. I can see what happens if I quarantine, if I immunize a fraction of people, how many people do I have to immunize in order to bring this down to a number of beds of 30, which my hospital can handle. I can ask what happens at the hospital between quarantining and not quarantining, between ensuring that nurses wear completely protective equipment versus not doing that. What are the sort of differences that I can expect in terms of this disease spread? So I said you can look at the spread of disease using agent-based models, and these are realistic in the sense they include geography, family structure, schools and hospitals, etc. These are very intuitive to work with. That is, for people who think about these you can think about an agent as a real person. I can say that this person is different from that person, is different from that person, and it's up to me to introduce the level of complexity that I want to describe a particular agent. But the difficulty is that you must customize each model. You must know roughly where people are saying, where are the populations, what is the geography, where are the schools, where do people move back and forth. So that's the expense. There's a lot that is loaded onto the model. But the nice thing is it's not a simple model anymore. So I can ask much more detailed and quantitative questions about the model. And as I understand more about the nature of the disease, the natural history of the disease in the individual, I can put that in. If I know there is a spectrum of susceptibilities in the population, I can put that in. All of these, I can work with models that are no longer as simple as the SIR model. It can simply be written down as a set of equations and solved. These are complicated models, and the only reason we can deal with them is because our computers are large enough that we can actually run 100,000 individuals over a period of a week with this level of complexity inside. So there is a trade-off, but the real nice thing about these is that you can put in information that is actually relevant and useful to deciding public policy in health. For example, preparing for disasters, preparing for epidemics, choosing between many different ways of dealing with the disease. Right? Is it targeted immunization? Is it shutting of schools? Is it quarantining? Is it just how much more vials of vaccine should I bring to that particular place? So these can be pushed in new directions that have not been used earlier. Okay, so that's the end of this set of lectures. I'll just quickly go back to this. Okay. So before I do this, let me just sort of summarize everything that I did for you. So this was a course nominally on the mathematics of infectious diseases, but if you were following carefully, I didn't do as much mathematics as I did general background to infectious diseases as well. And then I introduced the points at which mathematics enters this problem. That was a conscious decision because I could have done sort of various complicated mathematical derivations and proofs, etc. on the board, but they would have always been for fairly simplified models with none of the real structure of the questions that real clinical people or people would like to know about these diseases. So rigorous mathematical answers have their importance, but it's equally important to know when to go beyond that and how to frame these questions in different ways. And as good applied mathematician, you should be flexible enough to do that, to do both proper formal applied mathematical analysis, as well as use a computer simulation, try an agent-based model, try something stochastic. All of these should be tools that should be available to you. So we started off thinking about, we spent the first class basically defining various terms that were involved in disease, a little bit of the history of disease, where does disease come from, what is the difference between a viral disease and a bacterial disease, how do diseases spread, what are zoonotic diseases, what are vector-borne diseases, etc. We basically spent the first lecture defining various epidemiological terms. In the second lecture, we sort of went fully into proper models of the type of the SIR type. Let me just go back here and try and locate this. So we talked about the SIR model and related models, and I introduced the idea of the reproductive ratio at that point. The third talk was really about the SIR model and related models, but then we looked a little deeper into it. We asked, what do vaccinations do? What is the concept of herd immunity? And where do oscillations in these models come from? How do you have diseases that seem to go up and down in a seasonal manner? Then we did more complex models. We looked at age structuring in various contexts, age structuring in terms of childhood diseases. We looked at risk structuring in terms of models for high risk and low risk populations in the context of sexually transmitted diseases. We looked at stochastic models. We did Gillespie methods. We put space dependence in. Again, as I said, that was important, and looked at various examples, some of which corresponded to solvable situations and that we could actually make mathematical progress with. And then we talked about CA models, which really are numerical models. This last set of models here, again, this belongs to this class. It's just a more complicated SIR model. But with network and agent-based models, there's not much that you can do analytically. So these really come to much more the computational side of things. But they are a different way of thinking about this.